Right now, folks, we're just trying to focus on the ruler so we can get our measurement for our penny math. And so see how we can see all this granular stuff in the screen? That's the plastic, the ruler plastic. So now we've got our ruler set up, and these black dash lines are millimeters. So if we count the open space between them, we can see that the field of view right here is 3 millimeters. Okay, now we're going to work on the onion root tip slide. So we put the slide on the stage, and we're going to use these knobs on the side to move it into place. And we're actually not going to look through the objectives. We're just going to look at the slide and see where it looks like it's pretty well centered. Now we want to start with the stage in the lowest position, so we're rolling this towards ourselves. And now my trusty companion is going to do her best to get this focused by turning the coarse knob first till we can see something on the slide, and then we'll turn the fine objective to make it more clear as we get there. Okay, oh, we're still seeing, oh, I'm seeing something. Yay! Good job! Okay, let's stop here for just 1 second. Let's try to move this so it's more centered in our view. Okay, and right now, by the way, also, we are at 4 times on the objective, and then let's take a look up here. So our ocular is 10 times. So 4 times 10, we are looking at a 40 times magnification right here. And this is the onion root tip. And you can see here that the stain picks up the densest parts of the cell, which, wait for it, is which organelle? So that densest part is the nucleus in the center of the cells. And then we can sort of make out that there's this matrix of boxes around those nuclei, which are the cell walls. So let's see if we can get it up into a higher magnification. Now if we turn the objectives to 10 times magnification, we shouldn't have to move the stage. It should stay focused. Oh wait, that's oil immersion that way. So we're going towards the, yep, the yellow. Alright, let's see if we can get that focused. Oh, there we go! Beautiful! Okay, now let's take a look again. Okay, so now we're at 10 times 10, 100 magnification, and now look, we can start to see that there's some kind of variation happening inside of each cell. What is that variation? These pauses are so your mind can work. So what we're seeing here are stages of mitosis. Now, we'll get it up a little bit closer, and we'll think about how we can identify each stage. So, hold on a sec. Alright, see what happens if you turn it to the next objective 40 times. We're getting there. Okay, now try sliding the base plate with these vertical knobs so that's more centered, because we want the view to cover the whole screen, because we know the whole screen is 3 millimeter view. Okay? Alright, now let's see. Now with the fine objective, the outside of this horizontal tubing, see if we can get that a little more. Oh, so nice. So beautiful. Okay. Oh, we're looking at this. This is great. And sometimes if we roll through it, we can see different things because, right, to our eye, this is a razor thin, less than a razor thin layer of a plant, right? But the plant is not two-dimensional, it's three-dimensional. So when we roll through that fine focus, we can see different parts of the cell. So let me see if I can do this. Oh, I'll use this ruler. Alright, so let's look at all of these different cells. So think about here, pause the video for a second. You can take a screenshot of this video because this will come up in the next lab about mitosis. You could take a screenshot right here and try to find cells that are exhibiting all the stages of mitosis. So, give you a second. Think about that. Okay, so we were talking about the stages of mitosis, and the first stage of mitosis is prophase. And if you think about it, pro means before. So if we look over here, these are cells that are just in interphase. They're just resting, right? They're not doing a whole lot. The nucleus looks pretty evenly dense, right? But then if we look up here into this corner, see how we can start to see that the chromosomes are condensing, okay, right in this cell right here. So what that might look like to you is that there's variation in the cell. Okay, let's see, how about these ones, right? We can see that the chromosomes are condensing, prophase, all right? But once they're condensed, they've got to meet up in the middle, and so then we get to metaphase. And I like to call this metaphase. So the condensed chromosomes meet along the equatorial plate, right in the middle of the cell, and they start to organize themselves, okay? And then there's a lot of, one of the newest things we've started to understand in biology is how important the cytoskeleton is, and how there are reactions going on in what we think of as the interspaces. So essentially here we have more chromosomal organization, and the one phase I can't see on this particularly is anaphase, when these chromosomes would start to move towards the poles, towards the ends of each of the sides, so that the cellular contents are dividing, right? And then we get, after anaphase, into telophase and cytokinesis, right? And so if we look here, for example, see how these cell walls, it's like there's less mass per cell. It's not rectangular, it's more square. So that's like a cell being born, cytokinesis, okay? So you can see here how two cells, they still have the general same like biomass of this one large cell on the top, but then they'll continue to grow into interphase and become a large, or a rectangular sized onion root tip cell. So there's a quick lesson on mitosis. And now remember, the thing we're trying to figure out is how many cells fit across the surface of a penny. And so right now we know that this view that we're looking at right here in this screen is 3 millimeters. So it feels pretty doable, right? You could potentially count how many cells there are just by counting the entire population of cells in the picture. So it's not even a sample, just count every single one. But you could also come up with a strategy to figure it out. So you could figure out how many cells there are in the horizontal plane, how many are in the vertical plane, and then do some multiplication. All right, so that was the onion root tip. Okay, now we're gonna work on we're gonna use a blood cell, not a bone cell. And so we're just gonna reduce the height of the stage. We're gonna pull the stage down by rolling it towards us. And then we're going to move the objectives to the lowest power, because you always want to start your microscope in the lowest power. As we saw, once you go up in magnification, you can maintain that focus. And also, if you don't lower the stage, what sometimes happens is when you're turning these objectives, you crash into the slide and you break it. So now we're ready to take the slide out. Alright folks, so now we have some blood. This is frog blood, okay? This is frog blood on 10 times 4 magnification. And you know, we could just take a look, right? Like, we're just not seeing the same kind of organization that we could see before. See if we can get it a little clearer. Okay, now let's see what happens when we move it up in magnification. Alright, and here we are on 10 times 10, 100 times magnification. And notice how we're seeing the same kind of thing we saw in the animal cell, in that, or excuse me, the plant cell, that the stain that we use to be able to see these cells aggregates in the densest place of the cell, which is the nucleus. So we're just seeing a bunch of blobby circles with a darker dot in the middle. And the first thing you should notice, of course, is that plant cells have something that animal cells do not have. Ask yourself what that is right now. Alright, so plant cells have cell walls, but animal cells just have the membrane. Plant cells also have the membrane, of course. And so what we see here is that the cells take on a different kind of shape and a different kind of organization, right? Because also blood is not a tissue in the sense that it's not like making up a whole muscle. It is a tissue, however, but thinking about how blood is a fluid, right? Its purpose is to circulate throughout your body, and so that's why we see it in this fashion. But if we looked at a tissue like a muscle tissue, we would, or even a bone, we would see that there's a different kind of organization, but it is not rectangular like the onion cell because there are no cell walls in animal cells. Oh, this is so pretty. Okay, so here we are again, just like we did with the onion root tip. This whole view that you're looking at in the image on the video right now is 3 millimeters across in the view of the microscope. So like we said before, you could just simply count them, right? It's not too hard to count them in this screen. But if you were trying to figure out how many blood cells fit across a penny, you might also use some kind of strategy like counting how many are across the vertical plane. Like, you could literally, where'd my ruler go? Lay a ruler down, right? And you could count how many cells are lined up in that ruler face. And then you could place the ruler somewhere else, count again, place the ruler somewhere else, count again, and then average the measurements. And then you would know the average number of cells in the viewing window. And then you will extrapolate that view to 19.4 millimeters, which is the width of a penny, and find out how many of each of these cell types would fit across the surface of a penny. Alright, I think we have completed our lab here. We do want to, of course, leave the microscope in the proper situation where we're on the lowest magnification with the objectives, and we have the stage down in the lowest position. And away we go. Alright folks, so now we have some blood. This is frog blood, okay? This is frog blood on 10 times 4 magnification. And you know, we could just take a look, right? Like, we're just not seeing the same kind of organization that we could see before. See if we can get it a little clearer. Okay, now let's see what happens when we move it up in magnification. Alright, and here we are on 10 times 10, 100 times magnification. And notice how we're seeing the same kind of thing we saw in the animal cell, in that, or excuse me, the plant cell, that the stain that we use to be able to see these cells aggregates in the densest place of the cell, which is the nucleus. So we're just seeing a bunch of blobby circles with a darker dot in the middle. And the first thing you should notice, of course, is that plant cells have something that animal cells do not have. Ask yourself what that is right now. Alright, so plant cells have cell walls, but animal cells just have the membrane. Plant cells also have the membrane, of course. And so what we see here is that the cells take on a different kind of shape and a different kind of organization, right? Because also blood is not a tissue in the sense that it's not like making up a whole muscle. It is a tissue, however, but thinking about how blood is a fluid, right? Its purpose is to circulate throughout your body, and so that's why we see it in this fashion. But if we looked at a tissue like a muscle tissue, we would, or even a bone, we would see that there's a different kind of organization, but it is not rectangular like the onion cell because there are no cell walls in animal cells.